Second experiment to disprove this is the Louis Pasteur's experiment. Okay. He also did the similar type of experiment. That Louis Pasteur 1859 experiment is widely seen as having settled the question of a spontaneous generation. Pasteur selected several unique S-shaped flasks and divided them into two different sets. So this is the S-shaped flask, or this is also known as swan neck glass. Okay, see, this is the real glass used by the Pasteur. Okay, see, Pasteur prepared a meat broth. Pasteur added the prepared broth to the first set of S-shaped flask, boiled it, and sealed the flask with a lid. Pasteur added the prepared broth to the second S-shaped flask, boiled it, and uncovered it. Okay. Later, he placed both flasks, first and second sets, at a different location. Observations. After a few days, he observed that he noticed the dust collected on the neck of the flask. In the first set of flasks, there was no microbial growth. In the second set of flasks, there was a microbial growth. So, the flask which is covered with a lid. See, first jar which is sealed with a lid. In the first set of flasks, there was a no microbial growth because this jar is sealed with a lid. But in the second jar, there is a microbial growth. Okay. You observe that there is a microbial growth. Okay. You will see that both jar which is filled with a meat broth is boiled. Okay. Broth is boiled. Broth remains free of microorganism. Why they boiled the flask? Because if because of the boiling process, if there is any microbial content in this flask, they get died. Okay. So because of the sterilization process, because if this boiling, the flask get sterilized, which will support the experiment. Okay. So what is the result? He proved that the microbes are present in the air but will not arise from the air or a dust. In both sets of flasks, he uses the same boiled broth, but the first set of flask was sealed and the second set was left open for the microbes to enter, reproduce and multiply. So in the first jar, in the both jar, they used a same broth and also both the broths are boiled, but the first jar is sealed and the second jar remains open. Because of the opened jar, he observed that there is a microbial growth in the neck of the jar. Here, there is a microbial growth in the neck of the jar because the microbes is present in the air. Okay. This work was so conclusive that biology confined that the law of biogenesis, which states that the life only comes from the previously existing life. So these two experiments suppose the theory of biogenesis and also disproves the theory of abiogenesis. So do you understand the experiment of Louis Pasteur?
Mhm.
Can you recall it for me?
He tested what, like, where the microbes lived, and in his experiment, he found that microbes exist in the air and not in like, yeah, it's
So
same thing as the
he
last.
assumed that, so according to the Louis Pasteur, he stated that the microbes present in the air, okay, and these microbes are responsible for the growth of life in the flask which is opened, or we can say that not sealed, okay, and the flask which is already sealed, there is no formation of life. He explained that because the microbes present in the air does not get in direct contact with the broth in the in the flask which is already sealed. That's why there is a no any formation of life in the sealed swan neck flask, okay, but in the open flask the microbes get in direct contact with the broth. Because of that, they get multiplied, and you will see the presence of life inside that flask. Okay, understood?
Yeah,
Okay. So if we compare the abiogenesis with the biogenesis, that abiogenesis abiogenesis refers to the theory on the origin of life, describing that the life originated from the inorganic or inanimate matter. Inanimate means, can you tell me what is inanimate?
uh, like it it's not living.
Yes, a non-living thing. Okay. And biogenesis. Biogenesis refers to the theory on the origin of life, describing that the life originated from pre-existing living matter. So, according to the abiogenesis, substances proposed by Alexander Oparin, Stanley Miller, and Harold Urey, and biogenesis theory proposed by Theodor Schwann, Matthias Sladen, and Rodolph Virchow. Abiogenesis states that the life on Earth is originated from a non-living compound. It states that the life on the Earth originated from the pre-existing life forms. It is not scientifically proved by scientific experiment, based on the observations and the natural thoughts, based on the practical experiment and a material evidence. But in the next topic, or in the as we move further in this chapter, you will see that how the pre-existing life first forms, and you will see that these pre-existing life, or the first life, is no doubt formed by the help of inorganic matter, where we will study about the Operon and Haldane experiment, that the first life is formed because of the inorganic matter or the abiogenesis event. But these abiogenesis events are ridiculous, and these experiments, these theories are not any scientific support that the mud of the Nile River give rise to the frog, snake, or crocodile, or the sweet soaked dirty shirt, or the wheat brown helps in the formation of mice. These are not the scientific theories, but you will see that somehow this abiogenesis theory scientifically Alexander Operon and Haldane experimentally proves that how the abiogenesis or the first life developed because of the inorganic matter. Okay, So you have also another homework that you have to study about the experiment Miller and Urey experiment. Okay, so study about this experiment, Miller and Urey experiment. Okay, and in the next session we will study about the Big Bang theory. After that, the biochemical origin of life, and in the biochemical origin of life, we will also study the Miller and Urey experiment. Okay, so this experiment supports that the first life is formed because of the inorganic matters, that is methane gas, nitrogen, or the lightning spark. So, have you studied about this experiment, Miller and Urey experiment, in your classroom?
No.
No. So, what are the topics you have studied in your classroom in the evolution?
In evolution, we just started, but we studied like natural selection and like variation of traits due to selection pressure.
Yeah, those are the later topics we will study in this chapter. Let's see. In the starting of our formation of Earth, there is in the atmosphere, we have already seen that there is a methane gas, ammonia gas, water vapor, and H2 in the atmosphere, and uh, there is a continuous lightning happens on our Earth. So because of that, there is two electrode which continuously discharge a spark, and there is a condenser which cools the water vapor which is present in this circular glass, and you will see that there is a formation of water droplets, and these water droplets is collected here. Okay, and after some time when these collected waters get studied, there you will see that there is a formation of a prokaryotic cell, prokaryotic cells, or also primitive prokaryotic cells, which is known as conservates. So you can say that this is the first life, or we can say that the first cell which is found on the earth, conservates. Okay, so by the help of this experiment, that using inorganic material, that is methane, water vapor, hydrogen gas, ammonia, and the lightning spark, there this experiment shows that there is a formation of a life form or a formation of a cell. So the first life forms developed because of the abiogenesis from the non-living matter. Okay, so make a note or do more research on this experiment, that Miller and Urey experiment. And after that, I will also teach you these experiments, and then you will study, then you will understand that how the first life originated on our earth.
Yeah.
And after that, we will study the process of evolution.